Hi, everyone. So I think we're going to get started. So thank you all so much for joining. I'm so happy to see you all here, and I'm very excited for today's talk with Professor Alan Wolfe. I have seen the slides, and I can tell you that there's a lot of great content in there. Before we begin, for those of you who are new to our webinars, my name is Tamara Tobin, and I'm the founder of Urge Medical. Urge is a nonprofit that provides medical education to doctors and other healthcare practitioners on the topic of persistent and recurrent UTI and related conditions. We offer education through three means: live webinars such as this one, which take place about every 6 weeks, up-to-date scientific content on our website, and a private Facebook group for healthcare practitioners to communicate privately on the topic of diagnosing and treating patients with persistent and recurrent UTI and related conditions. I founded Urge so as to solve a gap in the market. With 25% of women worldwide suffering from recurrent UTI and 12% of women having symptoms of interstitial cystitis, which more and more research suggests could actually be undiagnosed infection, there is a very large patient population, yet patients are struggling to find clinicians who can offer novel approaches. Urge's objective is to share the most cutting-edge scientific research and medical approaches with doctors so that in turn you are able to help more patients and improve your patient outcomes. I will share details at the end of the presentation on how to join the Facebook group, and just a few housekeeping notes. If you have questions during the presentation, which I'm sure you will, please send them to me directly, and at the end of the presentation I'll be reading them out to Professor Wolfe. But without further ado, I am so excited to introduce our speaker for today, Professor Alan Wolfe. Professor Wolfe is a professor of microbiology and immunology at Loyola University Chicago Graduate School. He is the co-founder and director of the Loyola University Education and Research Collaborative and the founder of the Wolfe Lab, which studies the female bladder microbiome. He is the director of the Loyola Genomics Facility and is a fellow of the American Academy of Microbiology. These are just a few of his phenomenal contributions to the field of UTI research and education. And without further ado, Professor Wolfe, the floor is yours. And yeah, you were on mute.
Yep.
Oh, there we go.
Okay, so welcome. I'm a microbial geneticist and physiologist. I spent most of my career trying to figure out the circuit board inside of E. coli K12, the laboratory strain of E. coli, trying to understand how bacteria sense their environment and respond appropriately to it. And then, not quite 2 decades ago, I walked up to a urogynecologist, Linda Brubaker, introduced myself and said we had 2 words in common: E. coli. I study it, you want to kill it. She said, let's talk. And at some point during our conversations, she told me that some people thought the bladder was sterile. And I said, no flipping way. How could that possibly be? Have you seen the plumbing? Of course, she's the plumber. She's a urogynecologist, a surgeon that fixes the plumbing. So I guess I was being ironic. In any case, what we decided to do was to test the dogma that the bladder urine was sterile. And it was just about the time that next generation sequencing was becoming possible for smaller groups, which we were at the time. And so we could afford to do it. So what I want to do today is to talk to you first about why we thought that urine was sterile, a little history, talk to you about some of the diagnostic tools that are used for detecting urinary bacteria. I'm going to describe the urinary microbiome, what we call the urobiome. I'm going to talk a little bit about the difference between symptomatic and asymptomatic patients with focus on urinary tract infections and urinary incontinence. And I'll talk about whether urinary tract infections are always an infection, that is an invasion. And then I'm also going to tell you a little bit about some recent discoveries that we've made. So, right, you've already seen this. Here's the short story. So the old dogma is the bladder is sterile, that bacteria are not involved in most urinary disorders, that urinary tract infection occurs when a uropathogen crawls up the urethra into a sterile bladder, and it's very E. coli centric. But the new reality is that bladders have resident microbiota, right? Communities of bacteria that live in the bladder. Many of the urinary disorders likely result from dysbioses of those resident microbiota. Dysbiosis is a disruption of the normal community. And we have found that it's way more than E. coli. So why did we think the bladder was sterile? Well, let's go back to the key experiments done by Pasteur in the 1860s. Remember, he was trying to disprove the hypothesis that spontaneous generation actually occurred. So you know this. I mean, you studied it probably in high school. You know, he took a flask well, people took things like meat and put them in an open container and a closed container. And when it was closed, the maggots didn't come up. And so they said, well, spontaneous generation doesn't happen. But the people who were proponents of the spontaneous generation hypothesis said, well, maybe those little organisms, what we now call bacteria, maybe they spontaneously generate. And Pasteur did the same experiment. He took fluids, including urine, and put them in a sealed container and a non-sealed container without sterilizing. And he did the same thing with sterilized fluids. And what he found was the open container of urine that was sterilized or not sterilized became cloudy, became turbid. And the closed containers, whether they were sterilized or not, remained unclouding. Now, what he said was spontaneous generation didn't happen. Sometime in the next 20 years, there was a misinterpretation of those data. Those experiments were repeated by others. And Roberts is the one who started out a very, very key paper in 1881 that said fresh and healthy urine is perfectly free from bacteria or other minute organisms. The fact is that it was a misinterpretation of the data. The flawed logic assumes that urinary bacteria grow in ambient conditions, right? In oxygen. And what we found is that most do not. I'll show you some of that later. Skip ahead to the 1950s, and Ed Cass, who's an extremely brilliant man, he was an ID doc at Harvard. He wanted to know who was at risk of pyelonephritis. So he developed what we now call the traditional or standard urine culture protocol that clinical microlabs all around the world use. And it was hugely successful because it efficiently detected E. coli, which is the most common cause of pyelonephritis. Problem became when that test was moved to cystitis or bladder infections. The problem is that the standard urine culture test was designed to grow E. coli and other fast-growing non-fastidious bacteria, and it doesn't grow much of anything else. So the results came out that, urine was typically sterile, in quotes, right? No growth. Except, though, when it contained large amounts of E. coli or other fast-growing bacteria, that was labeled asymptomatic bacteriuria when there were no symptoms. So here's why we hypothesized the bladder was not sterile. So the first point was that we're now in the human microbiome era. We knew that there's one bacterial cell for every human cell, that we're actually walking, talking incubators. We can't live without our microbiome. They feed us and protect us. They they provide a substantial amount of the energy that we require to function. They feed our colonocytes, they feed the hepatic cells, they feed all sorts of human cells. And they protect us, right? They educate our immune system. They defend us against pathogens. They provide us with vitamins. They do all sorts of things, right? And the other thing is that we now knew that some organs that had originally been considered sterile were not. So a prime example are lungs. And the other piece of information was that we knew that most bacteria don't grow under the standard urine culture conditions. So let me talk to you about some of the diagnostic tools for detecting urinary bacteria. Right, I'm going to talk about the standard urine culture, or some people call it traditional 16S ribosomal RNA gene sequencing, an enhanced culture method that Paul Schreckenberger, the director of our clinical micro lab at the time that we did this work, called expanded quantitative urine culture, or EQUC. And I'm also going to mention multiplex PCR, and later on I'm going to mention a sequencing approach called shotgun metagenomic sequencing. So for detecting and identifying bacteria, that are really two different comparisons, right? So there are two culture dependent methods and two culture independent or DNA-based methods. So one of the culture dependent methods is the standard urine culture, or SUC, and then an enhanced urine culture such as our EQUC. And in clinical labs that do SUC and the few that do EQUC, they use MALDI-TOF to identify the bacterium that they've isolated. Now, I know that some groups that are now starting to do EQUC are using a sequencing approach to identify the bacteria that they're growing by their enhanced culture method. So the two culture independent or DNA-based methods are multiplex PCR and 16S ribosomal RNA gene sequencing. And I'll explain all of these in just a moment. So let's compare standard culture to EQUC. So the standard culture is what most of your clinical micro labs use. There are different variations on the theme. I'm going to focus on the approach that the Loyola Clinical Micro Lab uses and is used by many around the world. It's very good at detecting E. coli and other fast-growing anaerobic bacterial species, right? Those bacteria that can grow quickly at ambient conditions. Just throw it in a regular old incubator. The problem is it misses the majority of urobiome microbes. EQUC, which was designed by Paul Schreckenberger, as I'll describe later, it detects everything that SUC does, but also a lot more. It detects uropathogens and commensals, and it can assign it taxonomically to the species level. It's slower and more expensive than the standard culture, but only a little bit slower and only a little bit more expensive. But primarily it's good for research and when the standard urine culture consistently is negative in the presence of symptoms. So it's not particularly great for sporadic UTIs. It's really much better for someone who's got chronic problems like urgency urinary incontinence or recurrent UTIs, especially when the standard culture consistently comes up negative. So here's another way to compare it, right? The standard culture is blood agar plates, McConkey agar, two different conditions, incubated aerobically for 24 hours, and it detects most of the common uropathogens. EQUC is lots of different media. It's incubated under several different conditions. It's 48 hours, and it picks up more than uropathogens. Now, multiplex PCR does not require growth. It's detecting the unique pieces of DNA in the species that you're attempting to identify. It's focused on a subset of suspected uropathogen species, and currently at least the multiplex PCR panels that I know of don't detect commensals. It's pretty good for rapid diagnosis. The slowest part of it is getting it to the lab. 16S ribosomal RNA gene sequencing is the most comprehensive of these tests. It also doesn't require growth. It detects most of the bacterial taxa, right? But it does so typically at the family or genus level. It's not particularly good for getting species level assignment. And that matters because there are members of genera that are pathogenic and other members of the same genus can be non-pathogenic. So Bacillus subtilis is used as a probiotic. Bacillus anthracis is the etiological agent of anthrax. You'd kind of want to know whether or not it's Bacillus subtilis or Bacillus anthracis. But it's not fast, it's expensive, it's mostly used for research, although some companies are offering it for diagnosis. I think that's a mistake because most clinicians have no idea what to do with all of the information that they're being provided. In fact, we don't know what to do with most of the information that's provided at this point. So multiplex PCR is done where you have primer sets. So this is for polymerase chain reaction where you primers hybridize to a unique section of a bacterial genome, and you amplify the region between the two primers. So you do that with multiple primer sets that amplify unique species. So DNA that's in the chromosome of bacterial A is unique from B, C, D, and so on. So when you use all of these primer sets to amplify the unique part of these bacteria in a mixed culture, like a mixed sample like urine, if you see the amplicon associated with gene A, then you know that the bacteria A is present. If you see the amplicon associated with B, you know that that species is present and so on, okay? With 16S ribosomal RNA sequencing, today using next generation sequencing, it's a little bit different. What you're using is universal primers that will amplify a portion of the 16S ribosomal RNA gene from all of the bacteria that are present in a urine sample or any other sample. And then so now you have a library of amplicons, and now you use next generation sequencing to sequence those amplicons. So why the 16S gene? Well, 16S RNA is present in all ribosomes. So every bacterium has it, and it's essential for function. So it can only evolve very slowly. And because it evolves slowly, it can be used to distinguish between different species. Now, the reason you don't get species level identification with next generation sequencing is because the type of sequencing that's done in next generation sequencing are short reads. And there's not enough information in that short part of the 16S gene. You're not sequencing the whole gene, which is about a kilobase, 1,000 nucleotides in length. You're sequencing about 250. And sometimes there's not enough information in the sequence to distinguish one species from another. But typically you can tell the difference between one genus and another. Okay, so let me talk about some of our findings. I'm going to take you back about 10 years and tell you a little bit about the first paper that we published where we showed DNA evidence of bacteria in standard urine culture negative samples that we had obtained where the urine had been obtained from the bladder. Then I'm going to tell you about the development of EQUAC, that enhanced culture method, which provided evidence that the bacteria that we had sequenced were actually alive. Then I'm going to show you what bacteria you expect to see in continent adult women, and I'll show you just a little bit of data on men. And then we'll talk about the difference between individuals that have disorders like urinary tract infections or urgency urinary incontinence versus asymptomatic controls. And then some more recent data where we showed that the microbiota are dynamic, at least over a short term. And what's not on here is I'll show you that we can detect bacteria in the tissue, in the urothelial tissue. So let's start with the beginning. This is where I discovered, remember, I'm a bacterial geneticist, right, and physiologist. So I discovered that there was this thing called transurethral catheterization, suprapubic aspirates, ouch. And I learned about the midstream voided urine, or the so-called clean catch. Well, we call it the not so clean catch. What we did was to take cath urine, suprapubic aspirates, midstream voided urines, vaginal swabs, and for the suprapubic aspirate, we had controls. We had skin swabs where the needle went in, and we had a sham needle stick that went in but didn't go into the bladder, right? We sent the voided urine to the Clin lab for a standard urine culture test. And what came back is that all except one of those urines, which is important, I'll tell you, well, if I have time, I'll tell you about that one, that all the results were negative. So the question was, can we detect bacteria in culture negative urines? So we used 16S ribosomal gene sequencing, which I told you already allows detection of bacteria without growing them. What we found was the aspirated urine resembled the microbial communities in the aspirated urine, resembled the catheterized urines, which often didn't resemble the voided urine, which way too often resembled the communities in the vaginal swab. The aspirated communities did not look like the skin swab or the sham needle stick, so the controls worked. And we came to the conclusion that since the aspirate bypasses vulvovaginal contamination, that the bacterial DNA that we detected was indeed from bladder urine. We also determined that since the aspirate and catheterized urine resembled each other, that we could use catheterized urine for the study of bladder bacteria. So now we had evidence that that there were bacteria in bladder urine, but the 16S sequencing can't tell you whether they're alive or not. Now remember that these were culture negative. These were standard urine culture negative urines. So I looked at the genera that we could detect by 16S sequencing, and I said to Paul, well, Paul, these are not unculturable. They just don't grow under the standard urine culture conditions. Can you design a urine culture test that will allow us to grow the bacteria that we can detect by sequencing, but the standard urine culture test did not? Luckily for us, he could do that. And we had a master's student just starting out by the name of Evan Hilt, who could establish EQUAC in the lab. So let me show you that first. Let's show the standard urine culture to expanded quantitative urine culture. So SUC is 1 microliter of urine on 2 different types of media grown in ambient or aerobic conditions and incubated for 24 hours. For EQUAC, what we did was to increase the amount of urine by 100-fold. So we put down 100 microliters. We use multiple different media compositions, multiple different atmospheric conditions, and we let them incubate for twice as long, 48 hours. So let me show you evidence that standard urine culture is inferior to EQUAC. So this is a blood agar plate that was streaked with a urine, 1 microliter, right? 1 microliter, and it was incubated for 24 hours in ambient conditions in a regular old incubator. Now, what I'm going to show you is the same urine, 100 microliters on a blood agar plate, and it was incubated for 48 hours under aerobic conditions. So it's only one of about 8 or 9 different conditions that we do that constitutes EQUAC. This urine is not sterile. So what you can see is a bunch of big colonies with different morphologies. But there are not enough of them that if you only plated 1 microliter that you would detect them. You only detect them if you use enough urine, right, 100 microliters. And if you look carefully, there's all these little pinpoint colonies. These are slow growers. If you put 1 microliter in, let it incubate for 48 hours, you would have seen these little pinpoints. But standard urine culture is red in too short a time for these slow growing colonies to appear. Okay, so now we know that the urinary microbiome, or what we now call the urobiome, exists. The question is what microbes do we detect in the bladder? And how do these communities change over time? So let's talk about a study that honestly went on for about 8 years. We only published it in 2020. And it was the lead author was another graduate student of mine, Travis Price, who you'll hear about some more. Evan Hilt went from being a master's student to a PhD student. And she was also an author on this study. What do the microbiota look like in a typical cross-section of continent adult women? These are our asymptomatic controls. So let me explain what we're looking at here. This is a histogram. These were all done by EQUAC, although we do have 16S sequencing results on most of these individuals. I'm just going to show you the EQUAC data. What you're seeing here on the y-axis is how many colony-forming units per milliliter of the total, right? So you're here, the x-axis are each of the patients. There's 224 of them. Okay, so this individual here, 100% of the colony-forming units were lactobacillus. By the way, this color blue will indicate lactobacillus for all of my slides, right? But if you look over here, this individual was mostly lactobacillus, but a reasonable proportion was this maroon here and some of this green up here. So the maroon is Gardnerella, the green is Streptococcus. You can see that Gardnerella and Streptococcus are also found predominating. Predominate means that it's the majority of the microbiome that we detect. Staphylococcus is relatively common. And here's Escherichia, right? E. coli. Note, these are all asymptomatic controls. So E. coli is detected in individuals that have no symptoms. The black over here are those that are negative. When we sequenced the culture negative, EQUAC negative individuals, about half of them using 1 mil, sequencing 1 mil, about half of them will be sequence positive. And if we sequenced about 20 mils, almost all of them would be positive. So just because the EQUAC is negative doesn't mean that their bladders are sterile. So the conclusion here is that very often one bacterial genus predominates, and most often it's Lactobacillus, Gardnerella, Streptococcus, some Staph, some E. coli, some Enterococcus, that's this white here. And then there are others that you've probably never heard of, but you'll hear about in a moment, and some mixtures. Another thing that Travis discovered is that Gardnerella is a bacterium of premenopausal women. It tends to disappear as they get older, and E. coli tends to become more common in postmenopausal women. That's an issue, especially for older women who are in care, right? And maybe they're non-responsive. E. coli is typical of some women who have no symptoms. So just because you see E. coli, you get a positive standard urine culture test, doesn't mean that that individual has a UTI. So here's some male data. So this was led by Peter Bachik, who at the time was a urology resident. He's now an attending at the Cleveland Clinic. We took catheterized urine from men, voided urine from men. Some of them were controls. Some of them had benign prostate hyperplasia. And what we found is that the voided urine often didn't resemble the catheter. So the other is that the voided urine tended to have larger biomass. There are more bacteria in voided urine. We believe from a number of studies and some of our own work that the voided urine is primarily sampling the male urethra. And you need a catheter to sample the bladder. And you can see that not only do you see more bacteria in the voided urine, but you often see different bacteria in the voided urine than you do in the catheterized urine, although sometimes it's the same. So here's some strep. Here, this is shifted over. The green is streptococcus, so you see streptococcus in the voided urine and streptococcus in the catheterized urine. The blue here is staphylococcus. You see that. This purple here is candida. So again, one bacterial genus often predominates, whether it's candida or strep, Gardnerella, sometimes Gardnerella, some staph, and very rarely lactobacillus. Remember, these are men. Okay, what are the differences between symptomatic and asymptomatic subjects, right? I've shown you mostly asymptomatic microbiota. So again, this is Travis Price, and he and Evan did a big study where they compared individuals with urinary tract infections and individuals that did not have urinary tract infections, and they worked with fellows, urogynecology fellows from our urogynecology collaborators. All right, so the old dogma, UTI as an E. coli problem. So urogynecologists spend most of the time working with older women, perimenopausal. All right, so that's who we're looking at here. And sure enough, the individuals who were diagnosed with a UTI, about 50% of them were E. coli was detected. But look at the rest of them. The other 50%, well, this orange is other UTI-associated microbes. We're talking Klebsiella, Proteus, Pseudomonas, things like that. But here are a whole bunch of organisms that are not typically associated with microbes. And then there were mixed. These are polymicrobial infections or dysbioses, but they would be called contaminated by the standard urine culture test. The controls note that E. coli is common in older women, even at greater than 10 to the 5 colony forming units per mil, which is the typical threshold, right? And what Travis found was that thresholds were very poorly associated with symptoms. When E. coli was detected in a control patient, asymptomatic individual, right? There were 10 to the 5 colony forming units per mil, just like in the individuals who had E. coli but had UTI-like symptoms. And that was true for most, although not all, of the other species that were detected. There were, you could not find a threshold that would tell you that this individual has a UTI and this other individual did not. Okay, so now that we know, UTI is an E. coli problem, but what I'm going to show you is that it's not just E. coli and that the standard urine culture is flawed, right? This is work led by another graduate student who's still with me, Bailey Hochstetler. And what you're looking at is the frequency of detection. So this is the number of times, the prevalence, right? The number of times that a particular bacterium is detected. And then these are the most commonly detected bacteria. We took catheterized urine and voided urine from the same individuals. And the gray here, catheterized, solid, voided is hatched. The gray is analyzed by EQUAC, and the black, either solid or voided, is analyzed by SUC. Okay, now I'm going to break this down. SUC was designed to detect E. coli and other fast-growing facultative anaerobes. So these are facultative anaerobes, means those are bacteria that can grow anaerobically, that without oxygen, but they can also grow in the presence of oxygen. So let's look at E. coli, right? So here's catheterized urine. analyzed by EQUAC and SUC. It's doing a pretty good job. SUC and EQUAC are no different. Here's voided urines, and here's SUC. Here's EQUAC, and here's SUC. And again, SUC does reasonably well. You look at other members of the enterics. So E. coli is a member of the family Enterobacteriaceae, or the enteric bacteria. The same family includes most of the known pathogens, right? Klebsiella, Morganella, Proteus, Citrobacter, which isn't on here, and Ruitella. A related organism that's not an enteric is Pseudomonas. And to be honest, if these are the organisms that are in a particular individual, SUC does just fine, okay? But let's look at a different group of microbes. Here are ones that SUC either fails to detect them or usually fails to detect. So these are mostly organisms you probably never heard of. Actinotignum shallii, it used to be called Actinobaculum shallii, got a name change. Aerococcus urinae. Here are Candida, Alloscardovia, Strep agalactia, that's group B strep. That's important, right? And yet SUC is missing it. And Streptococcus aeruginosus. You'll hear about strep anginosus a little later on. Actinotignum, Aerococcus, and Alloscardovia. But here's one I really want you to pay attention to, because it's one that you probably know about. It's Enterococcus faecalis. There it is. Now, if you talk to a clinical microbiologist and you mistakenly say, well, SUC doesn't detect E. faecalis, they'll go, of course it does. And they're right, it does. Problem is, it misses it most of the time. And E. faecalis is a known pathogen, uropathogen. Okay. All right. I didn't tell you, these are older women with recurrent UTIs. So whether or not this relates to, sporadic UTIs of young women remains to be seen. Although in our hands, you still have the same problems to some degree. Okay. Let's look at the difference between UTI, urgency urinary incontinence, also a little stress urinary incontinence, and our controls, our asymptomatic patients. So this is one of our earlier studies. Megan Pierce was a postdoctoral fellow in my lab, and she basically jump-started this work, right? She taught my graduate students how to do the work that she had learned how to do. So what we're looking at is a species accumulation curve. If you've ever heard of a rarefaction curve, it's the same thing. What we're doing is asking the question, when we take our first sample, how many unique species were identified? These are by EQUAC, but you can do it by 16S sequencing or multiplex PCR. So the question is, how many different bacterial species, unique species, were isolated? So for the non-UUI women, right, these were our controls. The first woman actually had about, I don't know, looks like about 6 unique species that were detected. Whereas the first UUI woman, looks like they had 1 detected. The second urine that came in, well, we probably only, it looks like with the non-UUI women, we picked up 1 new species. So now we went from 6 to 7, but we picked up 2 or 3 new unique species in the next urine from a woman diagnosed with urgency urinary incontinence. So what you're looking at is the trajectory. And the trajectories are different, okay? There are way more bacteria, unique species of bacteria detected in women with urgency urinary incontinence than women that are asymptomatic. And here what we're doing is looking at some of the most common bacteria that were, and particularly the ones that are detected in a significantly different percentage in the affected women from the controls, okay? So we're looking at the frequency of detection, how common is it? The black or dark gray is those that were common in the UUI patients. The gray, the light gray, are those that were detected in controls. And what you can see is here's Actinobaculum, now known as Actinotignum shallii, some actinomyces. Here's Aerococcus urinae, some Corynebacteria, Gardnerella vaginalis, Streptococcus aeruginosus, which is the most common species that we detect in UUI individuals. The most common species that we detect is lactobacillus. But I want you to highlight, I want you to pay attention to these two. Here's two different lactobacilli. Lactobacillus gasseri is more common in UUI patients than controls. Lactobacillus crispatus is more common in controls than in UUI patients. Okay, so all lactobacilli are not created equal. Lactobacillus crispatus tends to be a good guy, like it is in the vagina. It produces all sorts of antimicrobials. We just published a paper showing it produces a small molecule called phenolactic acid that kills the hell out of almost everything, including yeast, E. coli, Pseudomonas, multiple drug-resistant bugs, except it doesn't kill other lactobacilli. Okay, so now I'm going to look at it a little differently. These are pie charts, and we're simply asking the question, what bacteria were common in the different cohorts? So all I want you to do is look at the colors and just the patterns, right? So here are our controls. About 50% were negative. About 25% had lactobacillus in it. And here's your strep, your Gardnerella. Here's your mixed or your polymicrobial individuals, right? Look at the UTI group. It's the same UTI group that I showed you earlier. 50% E. coli. Here's Klebsiella. There's some Proteus in here. That's Proteus. But here's people with UTIs, with lactobacillus, with streptococcus, polymicrobial communities. All right. So you can clearly see the difference between UTI and non-UTI. But here are the urinary incontinent groups, and they look very similar to each other. Although in a study that we just submitted, the abundances are different. There's a lot more of these bacteria in UUI than SUI. But the communities look similar. They look different than UTI, and they look different from the controls. Okay, so let me talk about some more recent discoveries. Most of what I just talked about are a few years old. So first, I want to talk about the work that Travis did, not as a master's student, which is what I was talking about before, but as a PhD student. So he decided that he wanted to follow young women every day for several months. And this is the study I'm going to tell you about. So here's the design. It's called ProFum, and it's ProFum because he and a urogynecology fellow named Beerta Wolfe, not related, asked, what does the microbiome look like in young women before and after and during taking oral probiotics? So you've got the oral probiotics in here and placebo, and these women sampled themselves every day for 3 months. Okay, first of all, Travis decided that he needed to screen these individuals. They're not going to catheterize themselves every day. No IRB would let us do that, nor would we want to. They had to do the midstream voided urine approach, and so we needed to know whether or not they were giving us a clean catch. So he used a periurethral swab and to compare to a voided urine. Now I'll tell you, the probiotic study turned out to be a complete bust. No difference, and we never detected the probiotic in their voided urines. So this just simply shows that in the individuals that screened positively, they were able to tell the difference between the periurethral swab and the voided urine. And he asked very, very invasive questions. They filled out the questionnaire every day for 3 months. What he found was 3 different types of patterns. There was the lactobacillus dominant pattern. There was a pattern where lactobacillus predominant communities switched back and forth with Gardnerella predominant communities. And then there was a pattern where strep, staph, and corynebacterial ratios changed. Now, I'm sure you noticed things like the brown here that showed up every once in a while. Those disruptions are actually dysbioses that are associated with menstruation. Here's a woman who menstruated once every 3 months. Here's a woman who menstruated every month, and every month actinomyces showed up. That's the brown. The other thing was that vaginal sex disrupted the microbiome. So I'm showing you one individual who had this lactobacillus, Gardnerella variation, right? And but almost every time she had vaginal sex, that's the pink dots, the green streptococcus showed up. What we found was it was Streptococcus mitis, which is considered to be an oral bacterium. It was found in this woman's urine. It initiated another study where we signed her up and her male partner. I don't have time to talk about it, but it's clearly a dysbiosis. He was not infecting her every time he had sex. Okay, I'm almost done. Question is a urinary tract infection always an infection? The question is how do bacteria reach the bladder in the first place? You know, E. coli is modal, Pseudomonas is modal, Proteus is modal, they move. How does Enterococcus get into the bladder? It's a non-modal organism. How does Lactobacillus or Gardnerella get there? They don't move. I mean, they don't have propulsive devices, okay? If you find commensal bacteria in a UTI test, it is a sign of infection or an overgrowth. And if you are some test results always indicative of infection? Why? And which bacteria are not commensal to the bladder? As I mentioned, how do they get there? Okay, so this is a study that's ongoing, so I can only tell you a little bit, but basically we're recruiting women with UTI-like symptoms. We're obtaining a cath urine, and it's being sent to the CLIN lab. At the same time, that urine and a rectal swab is being sent to us. My lab enriches for E. coli, right? We let it grow overnight in growth conditions that E. coli likes. And if the CLIN lab tells us that, yes, they detected 10 to the 5 colony-forming units of E. coli, we do shotgun metagenomic sequencing on those, the rectal sample and the urine sample. And shotgun metagenomic sequencing doesn't involve amplification of anything. You're sequencing everything. So you get the human DNA, and you get all the bacteria, all the viruses, all the fungi. But if you sequence deep enough, you spend enough money to get enough sequence, you can get strain level comparisons. So if we see the same strain in both sites, the rectum and the urine, then we can conclude that this is evidence of an invasion, right? An infection. But if we see two different strains, if the strain that we see in the bladder urine is not detected in the gut, we know that's not an invasion. And what we have found with the first 3 individuals is 2 invasions and 1 clearly not an invasion. So some urinary tract infections, at least by E. coli, are dysbioses. It's a disruption of the normal microbiome, which means that E. coli was present in that individual at low levels. Okay, so last is the urinary. Now that we know the urinary microbiota exist, I can tell you I've never thought that the bacteria are growing in urine. Urine is a lousy growth medium. And besides, if the bacterium is in urine, it's on the way out. So we've been using urine as a proxy for bacteria that we're absolutely convinced are associated with the urothelial tissue. But the question is could we actually detect bacteria in the tissue, in this case in biopsies? In the interest of time, I'm just going to tell you that these were biopsies we got from a team at Wake Forest led by Steve Walker, that some of these individuals are asymptomatic women, adult women of all ages, and some of them were female and interstitial cystitis, painful bladder syndrome patients. And I'm going to tell you, we see no difference between the asymptomatic women and the affected individuals. And that supports work that we've published using urine as a proxy. Okay, So we sequenced and we also imaged. So this is a figure that simply shows us we used a lot of controls to make sure that the samples that the bacteria that we're detecting in the samples were not contamination. So the green are the samples, the blue are different types of controls. So we now know that we can trust the bacteria that we detect by 16S sequencing, and this is what we see. What we see is a lot of Staphylococcus. It's very common, it's very prevalent, and very often it's relatively abundant. We see a fair bit of Lactobacillus. It's relatively prevalent, and when it's present, it's often abundant. We see E. coli. It's very prevalent, but not abundant. We see Gardnerella. It's prevalent, but not abundant, and we see a bunch of other bacteria. Okay, what about imaging? So cytokeratin is indicative of the urothelial cells. It's part of the cytoskeleton, and it's this light blue. We're detecting the 16S RNA, not the gene, but the RNA itself, and it's red. The DAPI, the dark blue, detects the urothelial nuclei, and when you merge these images together, you see that the bacteria are perinuclear. That is, they're sitting around the nucleus. We have many other images, and we're in the process of doing confocal, so we'll be able to get 3D. That way we'll be able to tell for sure whether or not the bacteria are inside the urothelial cells or not. So the conclusions are that the lower urinary tract is not sterile, and that broaches the question, what is the meaning of significant bacteria and asymptomatic bacteria, right? E. coli is common in older women with no symptoms. So the presence of E. coli does not necessarily mean UTI. So what does a positive culture mean? I can tell you the clinicians I work say, if there are no symptoms, don't treat it. The problem is when you can't determine whether there are symptoms. Standard urine culture is inadequate, and I'm warning you, some of your data may be flawed, okay? UTI is more than just E. coli, especially in patients with recurrent UTI. And on a short-term basis, the urobiome is dynamic. On the long term, it looks relatively resilient. That is, those patterns were maintained over 3 months. And we can detect bacteria inside the bladder tissue. What I'd like you to do is think differently. You go back to the clinic or your research lab, I want you to think a little bit differently. The dogma is wrong. And even people that say, okay, we're going to do 16S sequencing on our samples still have the old dogma in their heads. I can see it when I review or read the papers. And I'm just the front man for a very big group of individuals. The bolded individuals are current members or collaborators. We're the Loyola Urinary Education and Research Collaborative, LURIC. You can find us at this website and Twitter. And so any questions you can ask Tamara. She's going to relay them to me. Yes. And I'll put this up there because she wants you to know more about URGE. Yes. That I'll take questions.
Thank you so much, Professor Wolfe, for this really, really interesting presentation. We have quite a few questions, and they're all kind of addressing different parts of the presentation. So expect to be sort of jumping back and forth.
Did I unshare my screen?
You can leave the screen.
Okay.
Up if you like, because that way it's.
Maybe I'll go back. We'll see. Okay, go ahead.
So just to clarify, is it the case that asymptomatic women have intracellular bacteria?
We think so. The last bit of data I showed you, some of those individuals are asymptomatic, and we saw bacteria in the tissues. I don't know about the image that I showed you, but we do have images of asymptomatic women. And I do know that some of the women that we sequenced and detected bacteria, they were asymptomatic. And mind you, I tend to believe the data because we're seeing the same organism that we see in bladder urine, but we're seeing different ratios, right? We see a lot of we see relative abundance of staph, more than lactobacillus, more than E. coli, right? And that's exactly what I expect to see. I would expect that some bacteria are more intimately associated with the urothelium, others less so. And so the ones that are less so are more likely to be found in the urine than the ones that are intimately associated. But that's a lot more work needs to be done.
Yes. Is there empirical evidence that bacterial cells that embed in the bladder tissue and form quiescent reservoirs replicate within the bladder cells and continue to further embed? Right.
So it's referring to work originally done by Scott Holcren's group at WashU, where they showed that uropathogenic E. coli invaded urothelial cells of mice. They did a lot of work on it and have found some evidence that this happens in women. The group at University College London, Harry Horsley, has shown that Enterococcus faecalis invades urothelial cells of humans. And there's a few other, a bit of evidence. So they're in there, and they slowly replicate, although most of the work that we know on the behavior outside, above and beyond, are they present, has been done with uropathogenic E. coli. And they're growing, they're slowly replicating. And at least in the mouse model, what happens is that every once in a while, they burst out of these little pods, little biofilms inside the cell, and they replicate rapidly in the bladder, and that's associated with urinary tract infection-like behaviors.
Mm-hmm. So understanding correctly, there are quiescent reservoirs, so there are
yes,
like intracellular bacteria in both symptomatic and asymptomatic patients, but the type of bacteria may be different.
Yeah, the data that our recent data suggests that that's indeed the case. Mm-hmm. And what I couldn't show you is, because we didn't do it, we own the 16S, the detection of 16S was universal. So it just says that they're bacteria. We don't know the identity of those bacteria. Mm-hmm. Future studies would be using probes that are specific to particular bacteria that we think are present, either by 16S sequencing or some other detection method. Mm-hmm. Mind you, I need to point out that we're not the first to show this. 2 or 3 years ago, the group at UT Southwestern also showed bacteria associated with the urothelial tissue. They did it by imaging, they did not sequence. So we've shown that we can actually sequence the bacteria as well.
Interesting. So jumping to a different topic, have you or other researchers looked at the strain level? So do the E. coli isolates found in healthy normal controls or in asymptomatic E. coli dominant individuals comprise the same strains?
Right, good question. There's a running debate over what makes a UPEC, what makes a uropathogenic E. coli from a non-uropathogenic E. coli, what is typically called asymptomatic bacteriuric E. coli. And to be honest, so far, no one's been able to tell the difference in terms of looking at their chromosomes, looking at absence or presence of genes, right? That is, pretty much every UPEC strain has an asymptomatic bacteriuric strain that's highly related to it. You can't tell the difference. There's some evidence that maybe the difference is in gene expression. We're now looking at about 300 genomes of E. coli that we've sequenced, and there is some evidence that there may be a few genes that are common in E. coli genomes that came from E. coli isolated from people with UTI-like symptoms relative to those who are isolated from controls. But that's an ongoing study. We need to validate that. I tend to think that if there are differences, a major part that's kind of typically overlooked is that every individual is different. One of the things I didn't say, but is the working model in my group is that what we're looking at is ecology. You know, if you didn't know if you didn't know the concept of a lion, right? And someone said, "Oh, you've never seen a lion? Go to the zoo around the corner. It's not a very good zoo, you know, and go look at the lion on the concrete slab in a cage. That's a lion." Okay. And I'd say, well, if you really want to know what a lion is, go to the savannah in Africa and find out that lions live in families called prides. They spend most of their time hanging out underneath trees in the shade. And every once in a while they get up and chase some animal down and eat it. And you'd have to ask the question, well, what does a lion do in its environment, right? It's about the lion, it's about its prey, it's about the food that the prey eat, right? So if you're just looking for E. coli, you're kind of missing most of the information. You really need to be thinking about the pathogen in the rest of the urobiome in the host. And that's where I think we're going. We're going towards, instead of looking for the pathogen and trying to kill it, it's understanding the ecology of the bladder and trying to fix the ecology, improve, try to take a dysbiotic bladder or an infected bladder and move it towards a healthy one. So the answer to that question is no one really knows the difference between UPEC and non-UPEC strains at this point.
So actually, this is a good segue to the next couple of questions we had, which are, you talked about the natural, you showed some pie charts that show that symptomatic and asymptomatic patients have different microbiota. So what are some ways that the microbiome can be rebalanced for UTI patients?
Good. Well, first of all, I want to clarify. We're looking overall. Many of the bacteria that are detected in individuals with UTIs or UUI are present in individuals that are asymptomatic. It's a matter of prevalence, a matter of abundance or relative abundance, and it's a matter of the total composition. So yeah, I'm sorry, repeat the end of the last part of the question.
The question was, how can we modulate the bladder biome to readjust it?
So I know that this is in the future, right? But what I really like to do is, we have an antibiotic resistance problem. Clinicians are supposed to be good stewards of antibiotics, but they also have patients that are miserable and they want to be treated, and clinicians do what they think they're supposed to do. They don't have a whole lot of tools other than antibiotics at the moment. There are some, I mean, there's methenamine, there's big doses of mannose, there's a number of other things that can be done for UTIs, for urgency urinary incontinence. There are other types of approaches. But it would be great if we can get away from using antibiotics on urinary tract infections, because it's one of the most common reasons for a prescription. I'd really like to save antibiotics for people who are going to die tomorrow if they don't get them. And so, how do you do that? How do you treat a patient who comes in and they're unhappy? They're very unhappy. So possibilities are other antimicrobials. I told you about phenolactic acid. I don't know if that could be used, but it kills multiple drug-resistant Pseudomonas and Enterococcus. So it could work when antibiotics can't. We've never seen a resistant organism to it, although it's possible. There are phages, phage therapy. We're doing a lot of work with bacteriophage. I know that at UCSD that they've got a whole phage set up there. We can't compete with them, but and there is an example of an individual who was going to die, and UCSD used their approach, and they actually saved this guy's life. And it wasn't a urinary tract infection; it was something else. But so phage therapy is possible. There's a lot more work that needs to be done. There's the idea of using transplants, like a fecal transplant, a urine transplant, where you'd instill through a catheter. That's a possibility. There are all sorts of possibilities. We know that estrogen does, in fact, improve a dysbiotic bladder towards one that's more homeo or eubiotic. So there are a lot of possibilities, but they're all not ready for prime time. And I know it's hard to tell a patient who's been patient to be patient, but they need to be patient. We're not. I really don't want people to jump in and start trying stuff out. We don't have enough information yet, other than the old dogma's wrong, and we need to work hard to figure out how to deal with the new reality. We now have the possibility of figuring out alternative ways to approach these clinical problems.
Thanks. Thank you so much, Professor Wolfe. How can practitioners access EQUAC testing, given that SUC is, the standard urine culture is so inadequate?
Yeah, there's only a handful of places that I know that do it. So Loyola does, but only for Loyola patients and only if requested. I know that it's that UCSD does it, because Linda Brubaker, who was my partner in crime right from the beginning, a few years ago picked up and left Loyola and went to UCSD and got them to do EQUAC. I'm pretty sure that I know that UT Southwestern, at least for research purposes, has gotten EQUAC up and running. I think Duke might be, but the problem is it's hard. What I would do is I would go to your local clinical microbiology lab and the director and hand them some of our papers and say, read this. I've got a patient who has a chronic bladder problem, whether it's urgency urinary incontinence or it's recurrent UTIs, and see if you can talk them into doing it.
So just to clarify, this is a protocol that any practitioner could take to any lab, and it could be
done.
The design is open source.
Oh yeah, no problem whatsoever. I'd like to see more clin labs do it. I can tell you're going to have a hard time convincing them. They can do it. They don't want to in most cases. Now that's not true of all clinical microbiologists, right? I mean, but there are cases where people have attempted to get their local clin lab to do it and have failed. There are a few cases where they've gotten it to do it. Here's the problem, and it's the same problem you have with 16S sequencing, is now you're going to get a list of organisms and you're not going to know what to do with most of them. And that's up to us. We're in the process of trying to figure out who's bad, who's good, and how they interact with the host. But if you can get EQUAC done and you find that, like I mentioned, there was one individual in that first study that was the standard urine culture was positive. These were urines taken on the day of surgery, so they didn't find out till the next day that it was positive. It was 100,000 colony-forming units, 10 to the 5 colony-forming units of E. coli per mil. And yet when we sequenced, it was 1% of the sequence reads.
Oh, wow.
90% of the sequence reads were Actinotignum shallii and Aerococcus urinae. And if you saw a result like that, either by EQUAC or by 16S sequencing, you might think maybe it's not an E. coli infection, right? Maybe the symptoms are being caused by Actinotignum and Aerococcus urinae, and maybe I should treat them for those. Now, the problem is they are famous for being antibiotic resistant. So if you're treating the E. coli, you may be selecting for the Aerococcus urinae and Actinotignum shallii. So I would argue that if you're starting to throw antibiotics at somebody, think carefully. It's better to have more information than less. But just because you see a bacterium, you see a staph, a strep, whatever, doesn't mean that it's a bad guy. You know, the bladder is not supposed to be sterile. Your job is not to sterilize the bladder. Your job, at least ultimately, would be to try to get the bladder to be more eubiotic rather than dysbiotic.
Thank you. No, that's really helpful.
Yeah, I'm just really, I just have to caution people. We don't have the Holy Grail here. We just know that the Holy Grail exists.
Right.
Now
but
we're
so
searching
for patients on the ground and practitioners on the ground, if EQUAC is not available, are there some workarounds that they can do with the standard urine culture, or are there other testing types that you think they could use for patients who are not responding to treatment based on standard urine culture, or are getting negative results with standard urine culture?
One thing you could ask the clin lab to do is to incubate the plates for an extra day. Mm-hmm. Remember those slow growers? Again, you got to have a conversation with the director of the clin lab. And I would only do it on individuals that have chronic problems. At this point, particularly for young women who are having their first urinary tract infection and hopefully their last, keep doing what you're doing. I am worried. I mean, I don't know. Nobody knows how many older women have recurrent UTIs because their bladder microbiome was disrupted by an empiric treatment of antibiotics when they were 20 years old. I suspect some of them. But until we do that study, it's just a suspicion. So again, what I'm telling you is, I want you to think differently. I want you to be suspicious of what you think you know, and I want you to try to get more information, particularly on those individuals that have chronic problems. Mm-hmm.
So a couple of questions here that refer to bacterial consortia. Is it possible that other pathogens are sort of piggybacking, so to speak, on E. coli?
Yes, it's possible. Like again, I said, we think we're dealing with, this is microbial ecology, right? So not only might be piggybacking on E. coli, we do know that E. coli piggybacks on other bacteria, right? So there's work done out of...
How do you mean piggybacking?
Taking advantage of the presence of another bacterium. So work done by Mandy Lewis, who was at WashU, is now at UCSD, and her, at the time, postdoc, Nicole Gilbert, they showed that co-infection of E. coli with group B strep or with Gardnerella vaginalis made the E. coli infection worse. And they think they know why. So E. coli, uropathogenic E. coli, binds to the urothelium through something called a type 1 pilus. Think of it as a grappling hook with a sticky end, okay? And that sticky end sticks to mannose residues. Mannose is a sugar that's attached to the surface of the urothelial cell, the umbrella cells. But that mannose residue is hidden by another sugar called salicylic acid. Well, group B strep and Gardnerella, if I got this straight, both make sialidases. These are enzymes that will remove the sialic acid, exposing the mannose. And so now there are more mannose residues available for the uropathogenic E. coli to stick to. And I think that kind of relationship is going to happen over and over and over again. Okay, that there are bacteria that are creating, that you may not even detect, right? That they're there and they're creating a better environment for a pathogen to cause symptoms. Notice I didn't say an infection because it could be a dysbiosis, right? The opposite is true, right? So we showed that the reason that we went after Lactobacillus crispatus and found phenolactic acid is because Travis Price noticed that whenever he detected Lactobacillus crispatus, he didn't detect E. coli. So we dived in and said, well, maybe that, they're not co-occurring because there's some sort of negative interaction. And indeed we found one, right? So there are a lot of interactions going on. And like any other ecological niche, what we need to do is find out who the founder species are and who the keystone species are. Because if you've got a dysbiotic microbiome and you know what the keystone species is, maybe if you just destroy that keystone, it might be a minor member, but a critical member of that community. And if you got rid of it somehow, or you diminished it, maybe the whole community would collapse. And now you can begin to restore a eubiotic or a good microbiome.
Unfortunately, it sounds like there's no way of knowing who is the keystone.
Not yet.
Yeah.
Not yet. No, it's going back. Like I said, we now know there's information to be learned, and that's what we're doing. It's going to take some time.
Yes. A couple of questions about, again, coming back to modulating the bladder biome. So some studies show that fecal transplant can reduce recurrent urinary tract infection. How does that work?
I don't know, but it could be direct where if this is an individual who tends to get invaded by E. coli or other gut bacteria, then it's an invasion, and that would be a simple explanation. But for individuals where that's not the case, then you have to be asking a question: how could the gut microbiome indirectly affect the bladder microbiome? And no one knows the answer to that yet. I think both are probably happening. Note that there's somewhere in the neighborhood of more than a trillion colony-forming units of bacteria per gram of feces. I mean, there are just huge amounts of bacteria in your GI tract, particularly in the colon. And what's in the colon, in the lumen of the colon, is being taken up by the epithelium of the colon, the colonocytes, and being put into the bloodstream. If you do a metabolomic assay, a study that is measuring the metabolites in a human being, a lot of what you see are bacterial metabolites that humans don't make, but their bacteria do. And that is almost certainly the foundation for why the gut microbiome is associated with psychiatric disorders. There is no reason in the world why the same thing can't be true, where the gut metabolites from gut microbiome is getting to the bladder and affecting the behavior of the bladder. Now, that's all hypothesis. I have no data to back it up, but I'm pretty sure it'll turn out to be the case. It's quite reasonable. So I think it can be direct or indirect.
So another couple of questions on biome. If L. crispatus produces phenolactic acid, which kills most pathogens, why do L. crispatus probiotics often fail to produce clinical improvement in the setting of chronic UTI?
Okay, so the first question I have is, how is the crispatus being delivered? If it's an oral probiotic, we did not detect any difference with oral probiotics we never even saw that probiotic, which was Lactobacillus rhamnosus, in the individuals, in those 8 individuals that we studied, or the 6 that were on the probiotics. Now it's a tiny sample size, I know, but I think that the probiotics are likely to do their best if they're actually put in the urinary tract, or at least in the urogenital tract. There's a group in DC led by Suzanne Groh. They're at MedStar, where they've used Lactobacillus rhamnosus and they've instilled it, right, using a catheter into neurogenic bladder patients, spinal cord injury patients. And what they found was that rhamnosus was well tolerated. We now have money from a spinal cord injury foundation to actually put Lactobacillus crispatus into neurogenic bladder patients. It's also funding our PLA phenolactic acid study and mouse studies. We're going to see whether or not we can improve symptoms. So I think eating probiotics may help you with digestive issues, but I don't think it's going to help too much with bladder issues. I think you need to deliver it to the bladder. Again, early days. I mean, yeah. I'm sorry, Tamara.
Yeah.
I need to make it clear that in a lot of these things, I'm telling you my opinion based upon my experiences in a lab. I think it's necessary to
clarify. Yeah.
You take what I'm saying with a grain of salt. And also, there are clinicians that have done this type of research, and there are people who should be asked for serious clinical suggestions. Linda Brubaker, Beth Miller, Beth is my co-director at Loyola, Naz Siddiqui at Duke, a handful of other people. They're researching it, and they're also clinicians, and so they can actually give you clinical suggestions. I'm a scientist.
Yes.
I can't tell you what to do clinically.
Understood. Someone is asking, when likely is it that we'll see the results of this research, like of the lactobacillus instills?
We'll probably publish in 2 or 3 years.
Okay.
It's a 3-year study.
I know we're running quite a bit over. I'm just trying to fit in the last few questions. Interesting question here. Why? So if bacteria like Enterococcus are commensal to the bladder, then how do they then become intracellular? And is that normal?
Okay. So Enterococcus faecalis is like a lot of organisms in our bladders. They're actually what's called an opportunistic pathogen. They hang out, they're part of the normal microbiome, and then something happens that allows them to bloom or induce genes that are normally suppressed, not turned on, that allows them to become pathogenic. So a lot of, like strep throat, the organism that causes strep throat is a commensal. Most of us are colonized by it, but only sometimes does it flare up and become pathogenic. Enterococcus faecalis is one of those organisms. And at this point, we don't know how Enterococcus faecalis gets into the bladder, because I told you it's not motile. Of course, lactobacillus is not motile too, and yet it's there. I don't know how it gets there. And I don't know what causes Enterococcus faecalis to cause symptoms. But it appears to do so. One thing that's interesting that Bailey Hochstetler has found is, so she's studying Enterococcus. Enterococcus faecalis in most niches in the human body is encapsulated. It has this polysaccharide coating that protects it against the immune system. The urinary isolates are unencapsulated, which means that they actually, the capsule would have the effect of interfering with invasion. So it makes sense that urinary isolates of Enterococcus faecalis are unencapsulated so they can invade. Again, we don't know very much about urinary Enterococcus faecalis. Most of the work that's done on this organism are from other niches.
Last couple of questions. So a male patient is showing E. coli and Enterococcus faecalis in sperm and urine tests. Are these present in male controls? I think you might have gone over this, but.
Well, no. I don't know. I can't tell you how often we see E. coli in males. I suspect that that E. coli is detected, but we haven't looked at males with urinary tract infections. These were all. This patient
is this is the partner of a symptomatic patient. So his partner keeps getting UTIs, but
yeah, it's possible that it's possible that the E. coli is a minor, typically a minor member of the female's urogenital microbiome, whether it's in the bladder or not. But it's also possible that the male is the carrier and he's infecting her every time. Mm-hmm. I can't tell you which is the case.
And then last question again.
Wait a minute, you said that the E. coli was in his sperm, in his semen.
Yes.
Then yeah, he's probably a carrier. He's probably infecting her.
Mm-hmm. Do you have any studies on the prevalence of biofilm in chronic UTI and how to overcome it?
No, but I know that some individuals have biofilms. So the quiescent reservoirs of E. coli are considered to be small biofilms inside the urothelium, but I think you're probably thinking about, biofilms on the surface of the urothelium. I know of at least one patient who has a congenital issue and she gets a really bad Pseudomonas biofilm. It's just green. And Beth Miller's been treating her and having some success, but I couldn't tell you how she's doing it. I don't know. And it depends on the type of biofilm. But I don't think that your typical urinary tract infections are, the gooey type of biofilms that most people think of. I think they're microcolonies in places close to receptors that are causing the bladder to respond negatively to the fact that they're there.
Okay, and last question, again, coming back to the bladder biome and how it can be modulated. So you mentioned that oral probiotics did not have any impact on the bladder. And then you suggested that vaginal probiotics might. So did you do any research on that? And can you expand?
Yeah, no, the research that we're doing will be in humans and in mice, but we don't have a lot of data yet. We did an estrogen study, however, where individuals used a vaginal estrogen cream, and that changed the urinary microbiome, or the bladder microbiome, and the vaginal microbiome.
Is this pre or postmenopausal?
These were postmenopausal women. Now, on the other hand, I can't speak about it because I reviewed it just recently. I think there's going to be some data out there. There'll be a report out there that might dispute what we found. I think there's a lot more research that needs to be done. Now that's on estrogen, and that's not probiotics, and so it's different. But I think that we need to find out so things like estrogen and probiotics have a possibility of working on some individuals. We need to learn two different things. Which individuals are likely to be successfully treated with a particular type of approach, and what approaches, how do you deliver, what's the best way to deliver the therapy? And it's really early days.
Understood. Professor Wolfe, I think we've come to the end of the questions. Okay. And I want to thank you so very much for your time in talking to us today, and in spending an extra half hour. And for those of you who've been able to see the slide, you can find out more information at www.urgemedical.org about persistent UTI, or in the general research section. And there's going to be another webinar in about 6 weeks, and that's probably the pace at which we're going to be continuing for the rest of 2022. But once again, Professor Wolfe, I want to thank you so much for taking the time, for sharing your research, and thank you also for all your important work in this space. There are many, many patients who need you. So thanks again.
Tamara, do you want me to stay on?
We will hang up and I will contact you separately.
Okay.
Thanks again, everyone. Have a nice afternoon, evening, morning, night, wherever you are. Bye-bye.